welcome to my YouTube channel. So in this video, we are going to talk about the urea cycle. And just like we described in the other video about the amino acid metabolism, and one of the end products of metabolism is ammonia. And we said that ammonia is very toxic to body cells, and therefore the body has a mechanism to convert the toxic ammonia into non-toxic urea and maybe release it in urine by the kidney. Now, just to have a quick recap on what we did. For example, I said that the amino acid metabolism or catabolism can occur in the muscle. So let us just have a quick recap. So for example, when you have alanine, we said that alanine can react with alpha ketoglutarate and are going to produce two products, that is the glutamate and pyruvate. Alright. Then the enzyme that catalyzed this reaction was the alanine aminotransferase. Alanine aminotransferase. And this is the transamination process. This is called transamination. Alright. And we said that the glutamate can be transported into the bloodstream, then into the liver. So we have our glutamate. And we said, let me do this very well. And we said that glutamate can be acted upon by an enzyme called glutamate dehydrogenase. So this enzyme is going to be called glutamate dehydrogenase. Now when we see dehydrogenase, we're going to have the NADPH. NADP is going to be converted into NADP and it's going to be reduced. Right. Then we are going to add a water molecule. And so what are going to have? We are going to involve the release of ammonia. We're going to release ammonia and alpha ketoglutarate. Where we said that alpha ketoglutarate can again move on to another transamination reaction, maybe with aspartate or maybe another alanine or maybe with other amino acids, right? So that you can form glutamate and all those other processes. Now, ammonia, we said, can be transported into the mitochondria, right? So we have our ammonia. Then, inside the mitochondrion, ammonia can react with the CO2 or in the form of bicarbonate ions. So you can have ammonia plus bicarbonate ions and plus some molecules of ATP, that is 2 ATP. Then we are going to form what? We are going to form a new molecule. This new molecule is going to be called carbamoyl phosphate. So this is called carbamoyl phosphate. Alright. Carbamoyl phosphate, this reaction is going to be catalyzed by an enzyme called carbamoyl phosphate synthetase 1. So it's going to be called carbamoyl phosphate synthetase 1. Alright. Now we're going to have the carbamoyl phosphate. Now carbamoyl phosphate is going to react with a molecule called ornithine. It's going to react with a molecule of ornithine. Then we're going to form a new molecule. Now this new molecule is going to be called citrulline. Alright. Now, the enzyme that is going to catalyze this reaction is going to be called ornithine transcarbamylase. Alright. That enzyme is going to catalyze the formation of citrulline from ornithine and carbamoyl phosphate. Now, citrulline is going to travel out of the mitochondrion. Citrulline is going to travel out of the mitochondrion. So that we have our citrulline. Alright. Now, outside the mitochondrion, that is in the cytosol, remember you know the malate aspartate shuttle where we transported malate into the mitochondrion, then we got out the aspartate. Now this aspartate is going to react with citrulline to form a new molecule. Now this new molecule is going to be called adeninosuccinate. So I have adeninosuccinate. Alright. Adeninosuccinate. Now the enzyme that catalyzes this reaction is called adeninosuccinate synthetase. So adeninosuccinate synthetase. Alright, we have our adeninosuccinate. Adeninosuccinate is going to be acted upon so that it's going to be cleaved actually. It's going to be cleaved and they're going to release fumarate. And we're going to form adenine. Alright. Remember fumarate, where do you see fumarate? In the TCA, right? In the tricarboxylic acid cycle. So this can be taken to the TCA. Alright. Now, adenine, the name of the enzyme that catalyzes this reaction is called adeninosuccinate lyase. It's called adeninosuccinate lyase. It's going to lyse adeninosuccinate. So it's called adeninosuccinate lyase. Or some books you may find it recorded as adeninosuccinase. Still the same. Now, adenine can then be acted upon by another enzyme which is going to release urea. Right? And urea, of course, you know the structure is very simple. Just see NH2, urea, H2N, and the double bond. That is our urea. So, urea. Then what are we going to form? We're going to form ornithine. Then we're going to transport it back again. And actually, it's called L-ornithine. Then the chlorotoluene, and also going to have the L-citrulline. Alright. Then the enzyme that catalyzes this reaction is going to be called adenase. Adenase enzyme is going to release urea and also a molecule of ornithine to get back and track with carbamyl phosphate from citrulline, and the cycle is repeated over and over again. So this is the urea cycle. Alright. Now, what is the fate of this urea? Urea can be diffused in the bloodstream. So let us see how we are going to take this urea. So, urea inside the bloodstream. So when it diffuses inside the bloodstream, it's going to be transported to the kidney. When it's transported to the kidney, then it's going to be released as urine, right? Yeah. So that is the first fate of urea. Another fate of urea is that once it is diffused in the blood, a portion of it can also diffuse to the intestine, the small intestine, where it can be acted upon by bacterial ureas. So bacterial ureas are going to degrade urea into CO2 and ammonia. So let me just write here. Bacterial ureas are going to degrade this one into CO2 and ammonia. Then this ammonia can again travel back into the system, so that we're going to have ammonia. this is going to elevate the level of ammonia in blood. And of course, we know ammonia is toxic. So when you have elevated levels of ammonia in blood, this condition is going to be called hyperammonemia. So I'm going to have hyperammonemia on this way here. Hyperammonemia. And patients with this particular infection are going to be treated by being given a drug called neomycin. A drug called neomycin. Now, what neomycin drug is going to do is that it's going to reduce the number of the bacterial ureas so that you don't produce more ammonia. You're just going to reduce the production of ammonia, right? So that you can counteract this particular condition. So that is the fate of urea. We said lost in urine by the kidney, then maybe to the intestines, but the bacterial ureas can break it into CO2 and ammonia. Ammonia can travel back in the bloodstream, elevating the level of ammonia. Condition is called hyperammonemia, can be treated by drug neomycin, which reduces the number of the bacterial ureas. Remember, it is the bacterial ureas that degrade urea into form ammonia, and therefore, when we decrease or reduce the number of the bacterial ureas, so we don't have more production of ammonia. And guys, in other cases, when you consume more proteins, when you eat a lot of proteins, you're going to produce a lot of ammonia. Now, when there's a lot of ammonia in our body, some ammonia can react with the protons, right? When ammonia reacts with the proton, what are you going to form? You're going to form ammonium ion. So, ammonium can then travel into the bloodstream. Alright. This is the brain. So, inside the brain, we have inside the brain, we have astrocytes. Okay. Astrocyte. So that's the astrocyte. So the function of an astrocyte is to maintain the blood-brain barrier, right? Regulate the flow of fluids in and out of the brain. So what it does, it has an enzyme called the glutamine synthetase. So we have glutamine, let me write it here. We have an enzyme called glutamine synthetase. Glutamine synthetase. So what glutamine synthetase does is that it converts glutamate into, it converts glutamate into glutamine. Of course, you know the difference between glutamate and glutamine. Glutamine just has one extra as compared to glutamine. Now, this reaction requires ammonium ion to convert it. So, we need an ammonium ion. So, when you have elevated amount of ammonium ion in our blood, then to mean you are going to convert more of glutamate into glutamine, and therefore you are going to have elevated amount of glutamine in the brain. The amount of glutamine is going to increase. One thing about glutamine is that glutamine is osmotically active. Osmotically active, can draw a lot of water by osmosis. So what is going to happen is that the blood, because you know you have a lot of water molecules everywhere. Water molecules. So when the level of glutamine increases, it means we are going to transport more or draw more of water molecules into the brain. See this? Now, when you draw a lot of water into the brain, what is that called? That is called cerebral edema. That's going to be called cerebral edema. Right, when you have a lot of fluids in the brain. And this is characterized by, number one, you're going to have increased intracranial pressure. Right? So ICP is going to increase. Then you also have another condition called comatose. So these are some of the conditions characterized by the cerebral edema. When you draw a lot of water, a lot of fluids into the brain. Alright, now in other conditions, the ammonium ion can be converted into glycine. So, ammonium can be converted or can lead to reactions that form glycine. Now, when you have a lot of ammonium ion, you mean glycine is also going to increase. Glycine, on the other hand, is also osmotically active. So, when it is osmotically active, you mean you are going to draw much water into the brain, then cerebral edema, just as we saw. Now, how do we solve this? How do we treat patients with cerebral edema? Now, patients with these particular infections, we are going to give them drugs such as the benzoate. And we also have phenylbutyrate. We also have phenylbutyrate. So these are the drugs used to treat cerebral edema. So what these drugs do is that they are going to young, they are going to bind onto the glutamine and the glycine. They are going to bind and draw them. So they are trying to draw them from the brain. So they are going to draw them into the blood, then to the kidney, and they are going to be released finally or excreted as urine. So that's how these drugs are going to bind onto glutamine. So yeah. Glutamine and glycine. Alright, so that's how we solve that. So, what is the meaning about the urea cycle? The regulation, right? So, how can we regulate the urea cycle? Now, remember this particular enzyme, the CPS, call it the CPS. CPS1. The carbamoyl phosphate synthetase type 1. So this enzyme is activated, actually allosterically activated by N-acetyl glutamate. So it's going to be activated by N-acetyl glutamate. So it's going to activate carbamoyl phosphate synthetase to react to more ammonia, the bicarbonate ions, the ATP from carbamoyl phosphate, so that you can have the urea cycle. Alright, then other activation is like the glucagon, the glucocorticoid hormones. So you can also talk of the glucagon. Let me just write them here. You can have the glucagon and the glucocorticoids. These hormones are going to activate the enzymes involved in the urea cycle, so that we can run the urea cycle, we can produce the urea, and then we can release them in the urine or maybe in stool. I said some ammonia can then diffuse also in stool. I think I forgot to say that. Some ammonia can also diffuse in stool, then can be released or excreted. Alright. Then we also have another regulation. Actually, I said these are activators. We are also going to have the protein levels. So, for example, when you consume a lot of proteins, when there are a lot of proteins in your body, you mean you are going to have more glutamate, more ammonia, then so you are going to run more of the urea cycle, right? So when the protein levels are low, then the urea cycle is going to be low. When protein levels are high, the urea cycle is going to be very high, and then we're going to produce a lot of urea, a lot of ammonia, and so on and so on. So thank you so much, guys. I think that's what we have for you for this video. Please, if you find this video helpful, subscribe to my YouTube channel, comment, and like. So let's meet in our next video where we discuss more about your biochemistry and physiological functions of various processes happening in your body. Thank you.